we'll move from humans to rodents just for a little moment. But Cecile already introduced a little bit of brain clearance, but I thought I'd do it one more time, but slightly differently, because we work with this sort of fluid flows in the brain, and essentially this is sort of an old concept actually. So in the most of the body, there are lymph vessels, and I like to start to introduce that using this book. This is a book from 1787 by a professor called Paolo Mascagni at the Royal School of Siena. And basically he was a pioneer in his time. He discovered more than half of the lymph vessels that we know in the body using new techniques at the time. And he had like local artists essentially draw the lymph vessels around the body. And you get this really impressive network of lymph vessels in the body. And the point of lymph vessels is to essentially clean out the metabolic waste in your body. The thing about the brain is that it doesn't have any lymph vessels. You won't find any sort of images like this of the brain in his book, because the brain doesn't have any lymph vessels. So the brain has to do something else. And this is the point of lymphatics and of brain clearance, and this is sort of what we try to work on a lot at Simulon. So the really sort of seminal paper in this field is this paper here from 2012, which essentially tries to suggest a new, like an alternative pathway other than lymph vessels for the brain to clear its waste. So the point here in this paper is what they show is that essentially the flow of fluid in the brain is from arteries. You have these perivascular spaces, these spaces that surround blood vessels, and solute will enter through the arteries, and they will sort of go across the interstitium by, here there's a convective bulk flow. You can question whether that exists, but as these go into veins and then go out through the perivascular spaces along veins, and then it will pick up metabolic waste on this way. And another part of this sort of system is what's called aquaporin 4 water channels, which essentially are taught to mediate a bit of how this transport happens. Exactly what sort of role they play is still sort of not poorly understood. But we see that if you suppress the gene that creates aquaporin 4 in rats at least, they have less brain clearance. And altogether, this is what we call the glymphatic system. But the glymphatic system has a lot of questions. For example, what drives the transport along blood vessels? Is efflux really only through veins inside the brain? Does the flow, a convective flow over here really exist? And what's the role of, for example, capillaries in the middle of the brain, sort of maybe it's creating some loss of metabolic waste in this way? And in addition, many studies have shown that sleep seems to affect how efficient this clearance mechanism is. But lymphatics is really about what happens inside the brain. But then sort of the brain tissue will get rid of its solute, and then it has to get back into the body. And that's the role of what's called dural lymph vessels. So that's these two papers. In about 2015, they discovered, or they claimed to discover, that actually in the membrane that surrounds the brain, there are lymph vessels. And these would be essentially, so the brain would clear from inside the tissue, go into fluid, and then through these lymph vessels and go back into the rest of the body. And what's interesting is that, while this was new in 2015, you will also find this in this book from 1787. So lymph vessels like this are known, and they're known for a long time. They're sort of forgotten and then rediscovered. So a lot of what we look at when we do studies is these images, where they essentially inject tracers in humans or in animals, and then they take several images after each other, and then they track how these tracers move. So here is an example from a human. We take a pre-injection image and then multiple images after each other, and you can sort of simulate how the tracer moves. In rodents, sort of the way you do this is either you inject something in the back of the brain. There are sort of these fluid spaces in the back of the brain. But those experiments are a bit difficult because you don't know where things are going into the brain and where they're going out. So sometimes people will also actually inject deep into the brain. Of course, this does not work in the human. But that's sort of the way that we typically study this. And then we can look at how the tracer evolves and try to say something about parameters of this fluid flow. So now I'll show an example using actually data-integrated modeling, where we got experimental data from collaborators in Copenhagen. So I'm going to show you using data from a previous work from 2023. I don't do any experiments. This is experimental work from Copenhagen, where they essentially do these tracer injections in the back of the brain, and they do it in two types of mice. They give them two types of anesthesia. One anesthesia that creates an awake-like state called ISO anesthesia, and one that creates a sleep-like state, which I call KD anesthesia. And then they use a type of imaging called SPECT, which essentially gives you, it looks at the radioactive decay of a tracer, and you get the direct linear relationship to the concentration. And what's really interesting is that if you look at the two types of anesthesia regimen, there are quite significant differences between how sort of quickly that tracer goes out of the brain. So in the KD anesthesia, actually the tracer will tend to enter brain tissue much more, while in the ISO anesthesia, it sort of goes more or less directly to the nose. So if you just look at the total concentration in the brain, the peaks are quite similar, but the efflux phase is different. And if you look at also deeper regions of the brain, for example, like the cortex, in the KD group, much more tracer will enter the brain and less in the ISO group, similar in the hippocampus and also in the striatum. So what we will look at is try to actually simulate what happens when the tracer enters brain tissue itself. So we use the same relatively, like you say, relatively simple model, essentially assuming that this is mainly a sort of diffusion and dispersion problem. I'm going to look at specifically the extracellular diffusion, which is relatively known, and the perivascular dispersion, how they compare, essentially how important are this dispersion transport along these vessels compared to the extracellular diffusion. So a bit of the problem of doing this kind of modeling is that often there is a sort of contrast between the exactness we can use in meshing and what we can get from data. So actually the resolution of SPECT cells is quite low to get actually the exact interface between the fluid and the brain tissue itself. So we try to exclude the region that we estimate to be mainly fluid, and then we create meshes from an atlas. So now our problem is that we want to sort of simulate what happens in extracellular space and perivascular space separately, but the measurement is only the total concentration. So what's actually interesting is to think about what is sort of the importance of this boundary condition. Like if I put a different amount of trace threat in the extracellular or perivascular space to begin with, what is the impact on the sort of transport that we expect? So I'll show two examples. First, essentially assuming that at this boundary layer, it sort of permeates at the same rate, so that there's really no like sort of membrane at the tissue CSF boundary, which would essentially be to set these two concentrations to be equal. Another that essentially assumes that all the tracer is entering through blood vessels. And then we essentially fit these simulations to data that we got from experimentalists. So first, if you assume that essentially everything comes in through these blood vessels, and you try to compare these perivascular and extracellular diffusion coefficients, you essentially end up with the same diffusion coefficients. In this case, you actually don't really need the perivascular the dispersion. So here the diffusion is enough. So if you instead assume that there is like very little of a boundary, that they sort of go in, the tracer goes into the perivascular spaces and the extracellular spaces in a similar amount, you actually find that the perivascular dispersion coefficient is much greater than the extracellular dispersion, actually so much that it's unphysical. You expect the relationship between the perivascular and the extracellular diffusion coefficient to be about 3. So you need another mechanism. So one such mechanism is to essentially add now a velocity for penetrating arteries. So we add essentially just a velocity term that points inwards, and then we try to fit the norm of this velocity. And if you now add that, you get a rate of perivascular to extracellular dispersion that makes sense. It's about 3. And you find a this velocity of about 1 to 3 micrometers per minute, which is quite slow, but also similar to what you actually find in similar studies in humans. So to just sum up briefly, so here I've shown just how we can combine actually biomedical data with finite element-based modeling, going from actually experimental data in a rat, and building a mesh, and finally essentially trying to fit it to the data. And that's all I have. So thank you.
So let's thank the speaker. Any questions from the audience? Jack?
So at the beginning you mentioned this controversy about whether there's actually an effective process in the brain, and I've been hearing about this about 10 years now, and it would be interesting to hear a little bit more about it, your thoughts on it. It's an interesting point.
I mean, it depends on studies and who you ask and who does the experiment.
Yeah.
And
so it's still very controversial at this point.
I think maybe in the space is sort of more diffusion. The question is like what happens sort of when, especially in the human brain, you have this like thin layer of gray matter, and there, probably penetrating blood vessels, you have some advection that goes into sort of white matter. You won't see this as clearly in sort of rodents because they don't have as much. They basically only have gray matter. So, but this convective flow from arteries to veins, I'm not sure if it's, at least we haven't been able to see it very clearly, sort of.
Roman?
Thank you very much. It's fascinating. In this presentation, I think you've shown only one results for only one individual.
Yes.
My question is, does your when you say you fitted the model to the experimental data, do you have similar agreement with other individuals?
Yeah, I mean it's 4 from each group, but I showed just the one sort of 3D example. That's one animal, but this is like 4 animals in each box plot essentially, and you get sort of similar at least results.
So I have a question, Andreas. So the rodent brain is a lot simpler to mesh than the human brain, for many reasons. It is a smoother thing. So how would you think about expanding the model to go across scales, as in going down to more like cellular modeling? Do you have any thoughts?
Like modeling the cells itself.
Yeah.
I mean, I guess in the rodents, the problem there is that they don't have the same sort of segmentation tools typically, so it's harder to get actually the brain itself. Often they just use the atlases, so you can just use the atlas directly, so you can more idealize models. But I mean, of course, the advantage is that actually the rodent brains are much more similar. You don't have the same sort of huge variation between individuals as the others. But so I guess then you would be more looking at just modeling very detailed atlases. That would probably also give you something that's more also accurate compared to like actual individuals than in humans.
Okay. Thank you. So I think we should thank the speaker again.